Are you able to hear me? No, yes. No?
Yes, sir.
Okay, good. So you're able to hear. So today we'll be starting with, I'll keep admitting the people who will come late, so no problem. So today we'll be talking about basis of biotechnology and a little bit about restriction enzymes. So biotechnology can be defined as manipulation of gene for the benefit of mankind. So if you, in a good sense, try to clone a gene and try to express it, so in a host where it was not present earlier. So that will be called as biotechnology or recombinant DNA technology. So, for example, the concept of golden rice, you might have heard the concept of golden rice. I'll keep sharing some my screen so that you are clear about what I'm trying to say. Actually, our division does not have electricity right now, and I'm also running on a 2G network right now. So that's why I pick up. So golden rice is the rice that is able to produce vitamin A. Not vitamin A, actually, it produces a precursor of vitamin A that's called as beta carotene. Normally, we eat the endosperm of rice. Okay, I have a small presentation probably that can help you understand in a better way. Okay, I'm sharing my screen now. So if you're able to see my screen here, you can see that what I'm talking about, this is the golden rice, okay, and this is the normal rice. Okay, you'll be able to see my cursor. Now the need for golden rice came because there's a vitamin A deficiency all through the poor and the developing countries or the underdeveloped countries. In the map, you can see here the red regions, mostly in Africa or Indian subcontinent, the Syrian region, plus many other regions, the red ones. They are at clinical stage of vitamin E deficiency, what we call as VAD. So scientists came with a proportion that staple food in these places is mostly rice. In Kashmir also we eat rice, right? Chinese people also eat rice. Most of the African countries they eat rice. So rice is the staple food. So, but it lacks this vitamin A or beta carotene. So what they did, they thought, why not put the genes from a different source and put it in the rice so that you have rice that has carotenoids or beta carotene. That was the precursor. When we eat beta carotene, it breaks into two molecules of vitamin E in our stomach. So that's a source of vitamin E. So there's a full pathway of this beta carotene synthesis. In the flowers, they are red because of this carotenoid biosynthesis. We call it as a carotenoid biosynthesis pathway. And you can see here you have precursor of beta carotene is lycopene. And due to the lycopene, tomato is red. The lycopene is the major component of a tomato carotenoid. So it's red in color. So precursor of this is your phytoene and then general pyrophosphate. So after this GGPP or general pyrophosphate, you had the pathway up till here, but it lacked the pathway after that. So what they did, they took genes from different sources: Pseudonarcissus, Narcissus, Pseudonarcissus, that's a daffodil. They took genes from beer, they took genes from Irvinia, and they expressed it in rice. So now, if you can see my, are you able to see my screen?
Yes, sir.
Okay, good. So if you see it's a rice grain here, and the outer region is the aileron region, and the endosperm is the white region that we usually eat. So rice does have a little bit of carotenoids that are in this aileron region that are outside, that's the outer region. So, but the rice is polished before it's transported or stored, because since this region is rich in fats, okay, fats get rancid with time. So what they do is that they, who's writing on the board? Anyone who is doing it, please quit. So what we do, we polish the rice, okay, before transport and storage. So any meager amounts of carotenoids or beta carotene that's in this region is removed, okay. So the endosome we get is lacks this beta carotene. So what they did, they expressed these three genes: PSY is phytogen synthase, phytogen research, and lack of beta carotene. This is just an example that how biotechnology works. So if I go to the next slide, you can see here you have a, so once they express these genes, you have a rice that produces these carotenoids or beta carotene, and you have this golden rice 1. But the level of golden rice, the level of beta carotene in golden rice 1 was very low. So what they did, they made golden rice 2. And this golden rice 2 was made after they changed phytoenzymes. Earlier, they took phytoenzymes from Narcissus, Pseudonarcissus, but now in golden rice 2, they took it from maize. Okay, maize, since PSY is the main regulating enzyme, once they express this in maize, it has a higher quantity of 23-fold higher amounts of beta carotene in it. So this is how biotechnology can help us improve the living and how it can benefit the mankind. So what we do here in biotechnology, we play with the DNA or the gene. And before going to the gene, you should know that how the mechanism or how our cell works, how these genes work. So you have a concept of central dogma of molecular biology that says that the DNA, this is the gene part, okay, it transfers its information in the form of RNA, what we call as transcription. You know it, right? And later, this information from RNA is translated into proteins. Okay, so this from RNA to protein is called as gene expression. If this DNA does not express into RNA, it will not make proteins. So again, DNA can also synthesize its own counterpart in replication. What happens? DNA duplicates.
Replication. What happens? DNA duplicates.
Now that process is called as replication. And also in some viruses, the RNA is a genetic material, but it makes a DNA from RNA with a process called as reverse transcription. And the enzyme is reverse transcriptase. So this is the flow of information, how this occurs in a cell. And this DNA is packed in chromosomes. Okay. And for example, a human cell, how many chromosomes it contains? 23 pairs, right? 46 chromosomes. In a small cell, that is 0.2 micron. The width of DNA is 20 angstroms. Okay, the width of a chromosome, the length of a chromosome is around 7,000 angstroms. So DNA is packaged, what you call as DNA packaging. This, if we stretch the DNA from one end to another end, it will be around, from one cell, it will be about around 1 meter. N number of chromosomes will give you 1 meter long DNA strand. Okay, so this 1 meter DNA strand is packaged inside a cell that is your 0.2 micron. So there's a process, I will not go in detail, it's called nucleosome model, that shows different lines of condensation in which a gene is packaged in a chromosome. I will not go into that. So now, you can have a look here, a short view. You can see my screen here. You can see that this is your DNA. Okay, that's your, oh sorry, that's your gene. Okay, that's 2 nanometers or 20 angstroms. 1 angstrom is equivalent to 10 to the power minus 10 meters. Okay, and 1 nanometer is equal to 10 to the minus 9 meters. Okay, so this DNA or the gene is wound around proteins that are called as histone proteins. Okay, there are many types of histone proteins. You have H1A, H1A, H2B, H3, H4, and then you have H5. So these histone proteins, the DNA is wound around this histone protein. Okay, and this makes around 110 angstrom diameter leads. Now again, they form a rosette structure, and they form this 20 or 300 angstrom rosette structure. Then they again condense in the form of a solenoid. Okay, so you can see here solenoid, that's around 3,000 angstroms. Then this condenses again and then into the chromosome, and the width of one chromatid, the one arm of one chromosome is called as chromatid. So one chromatid is around, say, 7,000 nanometer, and the total width of the chromosome is 1,400 nanometer or 14,000 angstroms. So you can see a DNA that is 2 nanometer, 20 angstrom small, fits how it fits in the chromosome. Then you have 46 of these chromosomes in a cell. So, and you see, if you stretch this chromosome in the homo DNA, this 2 nanometer strand, it will be as long as 1 meter for n number of cells, for n number of chromosomes, and 2 meters for 2 n number of chromosomes. Now, in biotechnology, we fiddle with this. We take a gene, we cut it, we put it in another organism. It's not easy. Okay, it's not easy, but we at SKUAST do it. Okay, we have a full-fledged facility. If somehow we can resume classes or practicals, if we get some directions from higher ups, then you'll be doing some practicals in which you'll be isolating DNA, you'll be isolating plasmid DNA, you'll be isolating DNA from plants, from fungus, from cells. Then you'll be digesting that with the restriction enzyme. Okay, and then you'll be transforming that into this all in your syllabus. You'll be transforming that into a host, right? So another example of how biotechnology can be beneficial for us comes from this example. You can see here. Okay, so there's a disease called as mastitis, which is highly prevalent. Okay, and in US alone, it's a loss of 1.7 billions annually, and here also in India also. So there's a causal organism called as Staphylococcus aureus, okay, that causes this mastitis. So what happens is this is the disease of the udders. This bacteria then spoils the milk that's produced by this cow. So now what scientists did, they took a gene that's called a lysostaphin. Okay, Okay, they took from as stimulants, and they express this gene in the udders of the cows. Okay, then once they express this gene in the udders of the cow, with the milk you have this now lysostatin protein also produced, and this lysostatin protein kills your the Staphylococcus aureus, and then you have the milk that is not infected with this. The milk does not get spoiled, and you have good production of milk also. So this is how also biotechnology can help. Okay. So apple is the crop of Kashmir, and we lose around 70% of this apple. Sorry, we lose around say 30 to 40% of apple to scab annually. It's a menace. The Venturia in equalis is the archaeological organism. There are many variants of this. Okay, sorry, there was an interruption. Some people are saying that the audio is not audible. They are not able to listen. Is this a problem with everyone or some?
So now it's okay.
So another biotechnological intervention, how biotechnology can help us. You can see here on the screen. I'll share again. So you have the scab menace that's around. Okay, we lose around 30 to 40% annually, and that's around say 17,000 crore loss. Okay, to prevent this from happening, we use 325 crore per year rupees of your fungitoxicants. So they pollute our environment, plus we are not able to prevent this loss. So we came in SKUAST, we came with the concept of cisgenic apple. So what we did, we know there are some resistant varieties of apple also. Okay, the wild types, okay, like Maharaji, it's also resistant. You have Kikichu that grows in Ladakh is also resistant. Malus Bakita, it's resistant. Malus floribunda, these are wild type, but they are not edible or they are not good to taste. Okay, the wild type apple, you see, there are hard skin. It doesn't taste good. However, the or delicious variety, it tastes good, but it comes to scab. So what we did a whole transcriptome sequence. It means we sequenced the number of all the genes that were getting expressed in this wild type. We also sequenced all the genes that we are expressing this delicious. Then we did a comparative transcriptome. Okay, so all the genes will be same. Mostly all the genes will be same, but there will be genes that will be expressed in the scab resistant apple that are not there in susceptible variety. That's why they are susceptible. They are succumbing to scab. However, the wild types that the scab does not occur there. So we were able to discover 40 genes that have a role that were highly expressive in these wild type varieties, and now we're trying to clone them to put them in the delicious variety, what we call it cisgenic. Okay, there are two terms. One is transgenic and one is cisgenic. Transgenic means, like for example, golden rice example I gave you. So there you are taking gene from maize and putting it into rice. So it's a transgene, a gene from one source into another. But here you are taking a gene from one resistant apple variety and putting it in a susceptible apple variety. So this is the same. The species and genus are the same. So here we will not call it transgenics, we call it as cisgenics. So we're trying to make a cisgenic apple here at SKUAST that is resistant to scab, and for that and it will save a huge loss that occurs annually to apple growing farmers. Plus burden of using these fungitoxicants every year, 325 crore. It's also come down, and the ill effects it has on the populations that are living besides these apple orchards will also go down. It has been shown that the areas where apple is grown, the population that are living in the vicinity have an increased incidence of cancers because of the overuse of these pesticides, these fungicides. So this will also come down if this we're successful in designing or making this systemic apple. Another how biotechnology can help us is that usually you see that in Kashmir we have a cold season. It's a prolonged cold season we have, and we have usually one crop per year. But in other parts of India or other regions, they have like two crops or three crops. So what we're trying to do here, we're trying to make a cold tolerant tomato that is able to grow in snow and that is able to grow in winter, that are able to grow in snow or in ice. So there are various genes that are involved in imparting cold tolerance. Okay, like you have if you go to Ladakh, you have a temperature of minus 40 to plus 40. Minus 40 in winters, plus 40 in summers. So it's a long temperature range. So, but there are plants there that grow that grow under that harsh environment. Also, like your Seabuckthorn, the Hypophyramnoids, Hypophytibetiana, these bushes, these plants grow there, and they are cold tolerance. So similarly, you have tomato varieties that are from the colder regions of the world. Like you have tomato varieties from Poland, you have tomato from Siberia, from Russia. So what we did, we have a huge repertoire of tomato variants here. Some we got from US, some we got from Russia. So and these they have this cold tolerance gene called as CBF, stands for C repeat binding factor. So now what we're trying to do, we have cloned this CBF gene, okay, in a vector, and we have this gene with us. So what we're trying to do is now put this gene in the varieties that produce, that has good production, that has good yield, okay, so that it also becomes resistant to cold. Now, you now know the concept of the central level molecular biology, how you have a DNA, then it transcribes into RNA and then protein. But not all the genes that are present in a tissue are expressed simultaneously. Okay, like for example, when baby is in the mother's womb, it does not have teeth, right? But when it grows, when it's born, then the teeth starts to grow. So these genes are called as they are temporally regulated. So these genes are temporally regulated vis-a-vis time, and some genes are there that are expressed at one part of the body but not at other part of the body. Okay, like for example, you can see here. Okay, so you have the eyes here, okay, and the nose here. The genes that are responsible for making the nose are expressed on in this region. Okay, the genes that are able, making an eye are expressing at the region of the eye. Okay, so what we call as, if you have studied a little bit of cell fate or developmental biology in your 11th or 12th, you must be knowing that you have a mad box gene, you have Hox gene, all those genes. So there are genes that regulate this. Okay. So now you have an eye where, for example, you have rhodopsin proteins that makes rods, or the protein that makes the retina of the eye, the proteins that make the lens protein. Okay, they will all be expressed in the eye, but they will not be expressed in the nose. Okay, so this type of gene regulation in which, or for example, the nails, okay, are there at the end of my fingertips. What if nails start growing from my nose? So that does not happen because they are regulated. So this type of gene expression is called as spatial gene regulation or spatial gene expression. That's vis-a-vis space. So you have two types of gene expression. One is vis-a-vis time, that's temporal gene expression that at One part of the time they express or they do not express, and the later part of time they express or do not express. For example, you see here it's an apple. You do not see that when you sow the seeds, the plant comes out, then you have an apple. No, it doesn't happen because this small plantlet cannot support this big apple. So there's a spatial, again, a temporal gene regulation here. So the plant is there, it will grow into a tree, and then it will start bearing the fruit, and then you'll have the fruit. Okay, at that point of time, this plant will be able to bear the weight of the fruit. Also, it will be able to produce new supply all the components of the fruit. So this type of expression is called as temporal, vis-a-vis time. Now, if you can see here, central dogma again. Now all the you have a DNA and then it's transcribed to RNA. Now all the RNAs does not code for protein, are not translated into proteins. Okay, there are many types of RNAs. The main are the mRNA, tRNA, and ribosome RNA that we have been hearing since we're born. Okay, mRNA, tRNA, and ribosomal RNA. So among these RNAs, only your messenger RNAs are translated into proteins. Rest stay as RNA, and they degrade as RNA. Okay, so there are different types of RNA that are also there apart from these messenger RNA, ribosomal RNA. You have snRNAs or signal recognition particle RNAs. Okay, they help in membrane integration. The transfer RNA, you know that they help in protein synthesis. Okay, then you have transfer messenger RNAs. Sometimes what happens when protein synthesis is occurring on ribosomes, so there's a halt. Okay, it gets stuck. Your tRNA gets stuck, so protein synthesis gets stuck. So these transfer messenger RNAs come, they rescue this, they open this stress so that the protein synthesis can continue. Similarly, there are RNAs involved in post-transcriptional modifications or DNA replications. You have small nuclear RNAs, you have small nucleolar RNAs, you have yRNAs. There are many types of RNAs are there. Then you have the RNAs that silence your gene. So once your RNA gene transcribes a messenger RNA, then this messenger RNA will make protein. But before that, another RNA will come and bind to this messenger RNA and silence it. Like you have antisense RNA, you have siRNA, you have shRNA, short hairpin RNA that's found in viruses. So these kind of RNAs, they silence gene expression. Gene is expressing, but they're stopping, they're halting the expression of the gene by binding with their messenger RNA. So this messenger RNA can no longer translate into a protein. Okay, so there are around 56 types of different RNAs that you should be knowing. I can share this presentation with you. Okay. So now on how these genes are expressed and how does a cell know that which gene to express where and when to express it. So there is a switch with every gene. Every gene has a switch. So the gene here, we are talking about a set of nucleotides that code for a diffusible product. So if someone asks you a definition of a gene, it's a set of nucleotides that code for a diffusible product. So this set of nucleotide means it's the DNA. So your gene is on your DNA. Okay? Or your DNA makes the gene. So now every gene has a switch that which gene to express and which gene not to express. For example, in case of apple, the seed contains all the information of how an apple should be produced, but the genes will not be expressed there. All the genes will not be expressed there at that time. So what controls it? What controls it is your promoter region. So every gene has an okay. When you talk about gene, you have a 5' end, you have a 3' end. Then, for example, here you have, I don't know what will happen if this 40 minutes are passed. What you start again? CR? CR.
Yes, sir.
Yes, sir.
So we start again, we join again. Okay, that's a lot of work, right? So you have
Okay, sir.
a gene here. Okay, so the gene has a 5' end and a 3' end. So I'll be telling you what that means. So 5' always has a phosphate and 3' always has a OH group. If you So here on the screen, you are able to see my screen. So you have a promoter region. Okay, now on the 5' end, if you go up, it's called as upstream. And if you go down the 5' end, it's called the downstream. So every gene has a start codon that starts ATG. It's like +1 here. And anything, and the promoter region is usually upstream of this gene. That means it will be here somewhere. So this promoter is the switch of that gene that the gene should be expressed or not. And that switch is put on and off by various factors. We call them as transcription factors. That means if you there are genes that are to be expressed in the eye, you have the promoter there. Okay, and that promoter to that promoter, there will be transcription factors that will bind. So, for example, the transcription factor for lens protein will bind at the promoter of that lens protein gene, and then the promoter will say, okay, now I have to uh, synthesize the mRNA. So then it will be expressed. So if the transcription factors are not present, so that means the promoter will not will stay there silently and it will do nothing. Where the gene will be silent, the gene will not express. So what I want to say here is that each and every part of or every cell of your body contains each and every set of genes. For example, my eye also contains the gene that synthesizes nails. Okay, my eye cells also contains the gene that makes hair. My eye also has the gene that produces blood, hemoglobin. But those are not expressed there. So only those genes are expressed in a particular tissue that are needed there. And what determines the expression is your promoter and the transcription factors. So it's similarly that you have a switch, but you have to put on the switch.